Hello, I am Swati and we will be continuing the series of abiogenesis and biogenesis, the theory of spontaneous generation of life and theory of biogenesis. As I discussed in the previous series, the ancient view of the life. Now this ancient view of the life, that life can come from the non-living things, was shattered by the sophisticated experiments by the Pasteur and Rudolf Virchow. When the Pasteur was conducting his famous swine neck experiment, the German physician Rudolf Virchow was independently formulating a crucial biological principle. He was also doing independent experiments and he showed that the life can come from the pre-existing life only, and he gave a famous term Omnis cellula e cellulae, which was published in 1858, and it was a powerful statement against the spontaneous generation of the life. So with the work of these scientists including Redi, Lazaro, Pasteur and Virchow, the theory of the spontaneous generation of the life was shattered and the theory of the biogenesis emerged, which says that the life can come from life only. So these were the experiments done by the Pasteur. The breaking point, when the flask necks were broken, the broth was having the microbial growth, and in the previous slides, in the previous slide I showed you the innovative design of the swine neck which prevented the passage of the air. Thus, air can enter freely into the broth, but it provides or it traps the dust and microbes in the curve. So, it was the waiting game. The broth remained clear and sterile, free of any microbial growth, which demonstrate that air alone was not sufficient for the microbial growth to occur. It was concluded that microbes or the tiny mini living organisms are needed to give rise to the life. So, it was the end of the myth and the scientific triumph. If you follow the circle, there was a myth which was circulated and which was accepted for 2 millions of years, that there is spontaneous generation of life from millennia or from minute particles. But the discovery of the microscope and then the design of sophisticated experiment by various scientists such as Redi, Lazaro, Pasteur established a scientific triumph, and they laid the foundation of today's microbiology. So these were the pioneers which lead the groundbreaking research for understanding the infectious disease and cell biology. Now we know that these diseases are happening because of certain pathogens which are infectious agents or microbes. So they inspire for the new discovery. So the journey of the ancient belief continues to break down by the invention in the science. The invention of the microscope by Anton Leeuwenhoek laid the foundation work for the modern microbiology. So carrying forward with the Pasteur's legacy, it was an end of the era. So there was a conclusive end on the century-late beliefs and on the religious beliefs on the spontaneous generation of the life, transforming the biological understanding fundamentally. Biogenesis firmly was established as a theory, and it was widely accepted throughout the globe, throughout the various religious and philosophical ideologies. Now everyone believes that life can come only from the pre-existing life, not from the non-living matter. So it is also the foundation of the modern science, which is based upon the sterile techniques. Earlier there was no sterile techniques. People never believed that microorganism can cause disease, but now we know the importance of the sterile techniques. For example, when COVID emerged, the only preventive way was the sterile techniques, using ethanol, washing your hands frequently, because in modern era we know the importance of the microbes, their potential to cause a disease and their potential to be beneficial for us in many industries such as bakery, such as alcohol industry and cheese industry, dairy industry. So by these practices and by these understanding, we can save millions of the life and we can make millions of dollars or rupees by using them for our own benefit. So how do we understand biogenesis today? Because all the experiments that was listed are of 18th and 17th century. How much we have progressed with new advancement in the techniques. The term is coined. Now the theory is well established, and there is also a theory called cell theory, which is the germ theory, and it explains the continuity of the life. Like all the evolutionary progress on the earth across the billions of the years can be explained on the terms of biogenesis, how the life on the earth emerged after Big Bang theory, and after the Big Bang theory there was certain simple molecules such as proteins, RNA, DNA, they combined together to form a simpler life form known as prokaryotic life form such as E. coli, amoeba, which were easy to divide and reproduce, and these life form on the face of evolution gave rise to the more complex life form which is termed as eukaryotic life form, and then these eukaryotes were also single cell organism, but then they unite together to make multicellular organism and gave rise to higher organisms such as amphibians, trees, and today's multicellular organisms, all the mammals. So the life continues from the simple pre-existing life to the more complex existing organisms. So if we see the life's origin with the modern insight, we can define abiogenesis as the scientific hypothesis. This abiogenesis in the initial years is the evidence that is done on Miller's experiment that says that approximately 3.8 to 4 billion years ago, there were no living organic and inorganic molecule which combined together through natural processes such as lightning, such as volcano eruption, and these combination give rise to the simpler molecules, and there was a crucial distinction. This abiogenesis is very fundamentally distinct from the spontaneous generation. See, when we say that the life originated from the simple biological molecules in the beginning, we are not saying that it originated from the non-existing life. We are saying again the spontaneous generation, that life gave rise to the life. It was only in the initial phases when abiogenesis happened, but it is very different from the spontaneous generation. It was important only for the beginning of the life on the earth, not for the continuation of the life. The continuation of the life on the earth is only possible by the biogenesis. So it is incorrectly suggested that the ongoing and regular creation of the life come from the non-living matter, which was the spontaneous generation, but now we do not believe in it, and now we believe in the modern generation of the life. So this is the timeline of the life's origin, from emergence of the first life through abiogenesis billions of years ago. It has been 4 billion years that the first life molecule originated on the earth, but after that all the molecules, all the complex forms that you see today have been originated from the pre-existing life. So this extraordinary biodiversity that we witness today come from the pre-existing life itself. But how we can account for this diversity? It is because of the evolution. The nature was ever changing, the climate on the earth was ever changing. Earlier there was dinosaurs, but the earth was struck by meteors, or there was the gaseous change, there was volcanic eruptions, so they got extinct, and the micro environment of the earth changed. The microbiota, the flora and the fauna which was present on the earth, they changed and they evolved. There were mutations happening in their genome which gave rise to the form which was more suitable for the present world, and hence we see the evolution on the living organism. Like we can see the evolution in different strains of viruses, we can see the evolution in various apes. We also originated from the apes by the help of evolution, but it is not a slow process. It took millions of years to evolve. So let us understand how the paradigm shifts and the scientific revolution happened. How the Pasteur's discovery created boundaries while modern science explore new frontiers. The Pasteurian wall and the beyond. So after Pasteur's work, the spontaneous generation was a closed chapter. It was never discussed further, and every philosophical, religious and scientific world agreed on his work. So he created a clear scientific boundary. Life today arises only from the pre-existing life. And this raised the foundation of the synthetic biology. Now the modern synthetic biology explore the artificial creation of the life. We can alter the genome of the organism. You have heard the terms GMO, which is genetically modified organism. So we can design the organism with novel characteristic, because now we know that we can give rise to a new life form from the already existing one. We can alter the already existing one organism with the help of certain tools. These tools are the part of genetic engineering of the organism, and by altering the genome, we can make that organism better suited to the environment. For example, we can introduce certain genes in an organism such as plant, which can make it withstand osmotic stress, which can make it withstand drought, or we can introduce certain genes in plants which will increase their yield or which prevents them from the attack of the pathogen. You guys have heard of the term BT cotton, which is a cotton variety which is protective against a insect. This is a GMO. So these works laid the foundation of the synthetic biology and the original quest. Scientists are still fascinated and they still continue to understand how the life first emerged on the earth, because right now we are not able to create life from inorganic matter. Even in Miller's experiment, he could create the biological molecules only, the amino acid. He could not create the life itself. So we are still on the quest of creating the life by the molecules, not only on the earth but beyond the surface of the earth. So the study of the life beyond the surface of the earth is termed as astrobiology. The search extends beyond the earth and investigation of the potential life origin on the other planets and the moons. We are also exploring the origin of the life on the Mars and the moons, because we have seen the presence of certain inorganic molecules over there, which were similar to the molecules which were observed in the earlier earth. So can they have potential to give a rise to living organism, which can even be a prokaryote? If we find any living organism or potential biomolecules which can give rise to a living organism, then the debate will begin again. Can living organism come from inorganic molecules, or we need a living organism? So this cycle of questions will continue again if we find any living evidence beyond the surface of the earth. So why this matters in India and the world? What is the use of these theories, what we learned today? Is it only for the theoretical understanding, or it has certain applications? So our understanding of biogenesis certainly has application in the field of medicines and agriculture. It has paved the way for the medical revolution. Understanding the origin of life and the microbiology shapes modern medicines. All the development of the antibiotics, vaccine and the sterile surgical procedure that saves countless life is based upon the study of microbiology. Earlier there were no antibiotics, but with the growth of microbiology, antibiotics was discovered, such as the discovery of penicillin. The vaccines were discovered, and sterile surgical practices. All the surgical instruments are now either autoclaved or heat sterilized before use, so that there is no infection. Now we know that the surgical items can have certain pathological microbes which need to be removed before another surgery is being performed. So sterile methods are very important. They also have agricultural influence. The principle of biogenesis inform agricultural practices, crop breeding programs and sustainable farming techniques which are crucial for India's food security. See, the agricultural land is decreasing. Now all people are selling their agricultural land and they are moving to the cities. But our population is increasing. So there is an increased fooding demand. There is an increased demand of producing food. So this can be only met when we have increased in yield, and we can have increased in yield by following the principle of biogenesis and altering the pre-existing genome of the plant and making the plants yield more. So by the help of microbiology and genetic engineering, we can have GMOs which have better yield, which are better tolerant to drought and flood, which are better tolerant to the insects and the pathogens, and thus are helpful in having good yield and providing the food security to the growing Indian population. Also, it is paving the pathway to the biotechnology leadership. So, Indian scientists which are working in the field of microbiology, synthetic biology and astrobiology are making India as a leader in cutting edge biological research such as CRISPR-Cas technology, such as development of the vaccines. See, when the entire world was battling the COVID, India came with its own vaccine. We were able to develop our own vaccine because we have now biotechnology leadership and leading microbiologists in our country which are helping us in designing better vaccines. And it also helps in meeting our philosophical or religious views with science. It beautifully connects the ancient Indian philosophy about life's nature with modern science and methodology and discovery. The earlier scientific discovery and the surgical discovery were done in India. If you study the ancient Indian text, you will realize that Shushruta was the one who conducted the first surgery, and our grandmoms, our dadi nanis were very much familiar with the sterile technique. They never knew the term microbes, but whenever they were doing deliveries in the paddy fields, they were heating their इंस्ट्रूमेंट्स बिफोर कटिंग द अम्बिकल कॉर्ड. आप लोगों ने देखा होगा जब गांव में डिलीवरी होती है आज भी, अगर घर में डिलीवरी होती है वो अपना हसिया हीट स्टेराइल करते हैं. They do not know the technical term for this heat sterilization, but actually this heat is killing all the microbes which are present on their instrument so that newly formed mother or child को infection ना हो. So microbiology also tell us that our philosophy was rich. Our Indian knowledge system was rich in the sterile conditions also, and we were the ones who believed that life comes from the life. We were the ones who never believed in abiogenesis. We believed that all living beings are connected and life is one. So in that way we were on the same line with the ancient Romans, because they also believed that there is something in the air which is bad. They did not had any technical term for that, but they knew that there is something bad in the air which is infection or the microbes. And similarly within our ancient principle, we knew that if someone has chickenpox, we need to separate that person because that person is spoiling the air. There is something bad in the air. So that bad or that chhuachut was actually the microbes, and we needed more sterile techniques to evade that. So what we can conclude from today's lecture is how we travel from myth to modern science with the help of scientific experiments. So spontaneous generation was a compelling idea and it seemed to explain everyday observation, but it lacked scientific rigor. So it was powerful, but it was flawed. So anything that is flawed need to be questioned, and it can be only questioned by scientific manner and scientific experiments. So experimental truth will lead us from myth to the modern science. Biogenesis, proven through careful experimentation, stand as the cornerstone of the modern biological sciences, which include everything from biochemistry to biotechnology to microbiology. And it's a continued inspiration. The story of life's origin continues to inspire discovery, innovation and wonder across the scientific discipline worldwide. So the take home message from today's series is life comes from life. It's a profound truth that transformed the science forever and continues to guide biological research into the future. Thanks